Good morning. Welcome to our new webinar series, everyone. My name is Dr. Liceo Otero, Chief Medical Officer and Laboratory Director of LifeLens. In this session, we will talk about telomere length measurement and monitoring, and we will also discuss its implications in stem cells, therapeutic potential, and male infertility. For those of you who may not be familiar with our company, I'd like to make a brief introduction. LifeLens was founded in 2010 as a spin-off of the Spanish National Cancer Research Center, the CENIO. Our headquarters are located in Madrid. Currently, we are the world leaders in the field of telomere diagnostics. We are the only Spanish laboratory fully accredited as a PLAYA lab, and also under the ISO 15189 standard, which accredits us to provide our services in more than 65 countries worldwide. Our TAT is the only technology in the world that's commercially and clinically validated for measuring telomeres at the individual chromosome level. In case this is the first time you are attending our webinar or you have never worked with us before, I'll go through some of the most important aspects of telomere biology. Telomeres are structures located at the end of chromosomes. They are formed by a repetitive sequence of no-coding DNA, whose function is to protect the coding DNA. They play a very significant role in the aging process. You could consider telomeres as a biological clock. By measuring them, you can have a clear idea of how close is a cell to its senescence. Short telomeres are a contributing factor to virtually all age-related diseases. Every time a cell divides, its telomeres get shorter due to the end replication problem. This implies that every cell has a limited replicative capacity, with a limited number of division cycles. This number of cycles is known as the Hayflick limit, which goes from 50 to 70 replication cycles, depending on the type of tissue. After reaching this limit, cells enter into senescence and ultimately go into apoptosis. In this slide, we can see how the aging process is being driven in large part by telomere attrition. Every time cells divide, their telomeres get shorter. Cells' functionality decreases, and so does tissue and organs. All of this happens as part of the aging process, which may provoke age-related diseases. The correlation between telomere shortening as a major contributing factor to age-related diseases has been widely studied and supported by more than 25,000 publications. Short telomeres drive damage to the DNA into the cells, which affects tissues and organ systems, ultimately leading to system dysfunction and the occurrence of age-related diseases, like cardiovascular diseases, cancer, and fertility problems, among others. In 2012, Overall carried out a study where the telomeres of more than 800 individuals were measured. And as you can see in the graph, telomere length can vary from one individual to another at any given age. Telomere attrition rate is also highly heterogeneous, as indicated by the slope of the graph. This rate can vary widely. Even if you were born with shorter telomeres than average for your age, you can make changes that will impact your telomere attrition rate. This will decrease the slope, indicating that your aging process has slowed down, decreasing your risk of having age-related diseases. The ultimate goal of preventive and personalized medicine is to maximize not only lifespan, but health span. Now, what are the main factors impacting our telomere length? Genetics certainly play a partial role in determining telomere length. To this day, we can't do anything to change this. On the other hand, we also have environmental factors, how you live, where you live, what you eat. These play an even larger role, and you can actually do something about them, such as changing and improving your lifestyle habits and ultimately your overall health status. Now we will review the correlation and applicability of telomeres biology and measurement in stem cells research and male infertility. We will start by discussing telomere length measurement and its correlation and advantages on stem cells and IPSC'S research and potential clinical applications. Stem cells are a class of undifferentiated cells that have two major characteristics. Pluripotency, which means that they are capable of differentiating into different types of specialized cells, and self-renewal, the fact that they can divide in order to produce more stem cells of the same type. And there are two types of stem cells regarding to its origins. We have embryonic stem cells. They are isolated from the inner cell mass of blastocysts, and adult stem cells. They can be found in various tissues, but they are limited in their ability to differentiate based on their tissue of origin. In other organisms, stem cells and progenitor cells act as a repair system for the body, replenishing other tissues. In a developing embryo, stem cells can differentiate into all the specialized cells, but also maintain the normal turnover of regenerative organs such as blood, skin, or intestinal tissues. Induced pluripotent stem cells are differentiated cells that have been reprogrammed to an embryonic-like state by transferring nuclear content into oocytes or by fusion with embryonic stem cells. The expression of the OCT4, SOH2, KFL4, and semic transcription factors turns into the reprogramming of these cells, and they exhibit the morphology and growth property of embryonic stem cells, acquiring the ability to differentiate into specialized cells. Stem cells are currently being investigated for their therapeutic potential in areas such as regenerative medicine. Currently, the most used stem cell therapy is the bone marrow transplant, a treatment for some types of cancer like leukemia, multiple myeloma, or some types of lymphoma. The contribution of stem cells to cure neurodegenerative diseases have been unraveled and explored extensively over the past few years. Beyond substitution of the lost neurons, stem cells act as immunomodulators and neuroprotectors. A large number of preclinical and a small number of clinical studies have shown beneficial outcomes in this context. Today, stem cell therapy offers promising hope for almost all forms of neurodegenerative diseases, including Parkinson's disease, Huntington's disease, Alzheimer's, and amyotrophic lateral sclerosis. Spinal cord injury is a devastating condition, and for which there is no effective treatment, producing great personal and social costs. Stem cell transplantation is a promising therapeutic strategy, though much preclinical and clinical research work remains. Stem cell therapy offers several highly attractive strategies for spinal cord repair, including replacement of damaged neuronal and glial cells, remyelination of spare axons, restoration of neuronal circuitry, bridging of lesion cavities, production of neurotrophic factors, anti-inflammatory cytokines, and other molecules to promote tissue sparing and neovascularization, and a permissive environment for plasticity and axonal regeneration. Laboratory experiments and recent clinical trials suggest that cell-based therapies can improve cardiac function, and the implications of this for cardiac regeneration are causing great excitement. This therapeutic approach is likely a clinical reality that may revolutionize the treatment of cardiovascular disease. In somatic cells, telomerase activity is usually diminished after birth, so the telomere length is gradually shortened with cell division and triggers cellular senescence. But in embryonic stem cells, telomerase is activated and maintains telomere length and cellular immortality. It has been shown that telomerase controls tissue homeostasis and organism survival. This suggests that telomerase activity and telomere length can directly affect the ability of stem cells to regenerate tissues. When other stem cells are reprogrammed into IPSC'S, telomerase activation and telomere length extension occur. Furthermore, IPSC'S exhibited heterogeneity in the levels of telomerase and telomere length. The fundamental role of telomerase activity and telomere length play in the generation and functionality of IPSC'S highlights the importance of telomere biology in this field. In addition, the telomere analysis could be very useful in order to determine the quality and the differentiation potential of both stem cells and IPSCs. Senescence is a biological process, and we have the tools to slow it down, halt it, or even reverse this phenomenon. This scientific breakthrough could allow patients needing a regeneration therapy to have a fighting chance to slow down or even reverse the aging process to help further lengthen their lives. Stem cells are found in all humans and enable us to quickly recover from injuries. They provide the nutrients and instructions for a stronger and healthier body. All throughout the natural process, your stem cells actively work to help replenish your body. If some of your organs are damaged, these repair stem cells tend to the damaged parts of the body to begin repair. As people age, the production of your stem cells also diminishes over time. Stem cells are usually created and distributed from bone marrow, but over time the production decreases and it no longer releases as many stem cells as it used to. This reduction in circulating stem cells is the main reason why nagging injuries and illnesses take much longer to heal in old age. The fewer stem cells in your body means fewer cells capable of repairing and rejuvenating your body naturally. This has been the target of regenerative medicine for a long time, and now stem cell treatments are being used for anti-aging and to treat age-related diseases. Nowadays, the relationship between telomeres and stem cell therapies has been established, as in stem cells, telomerase is activated, maintaining telomere length. Going a little bit further, in combination with other factors and lifestyle habits, these therapies could help with telomere length maintenance and promote the slower shortening rates as they reduce overall frailty in an individual's body. Telomere testing could potentially be an interesting and useful reference point before and after a stem cell therapy, as this biomarker can provide information about the potential effects of the therapy by monitoring potential changes in the rate of telomere attrition. If you want to study more in depth the correlation between stem cell therapies effects or stem cells quality and telomere length, here we provide you with a comprehensive list of publications. Now we will continue with telomere length and its potential application in diagnosing of male infertility. Infertility has become one of the 21st century's biggest growing social problems impacting people both in developed and developing nations. Numerous studies suggest that between 8 to 13% of couples are infertile. This means that women cannot achieve pregnancy when trying to conceive for a year or even longer. Out of all infertility cases, the male factor alone represents around 30%, while the other 30% is due to a combination of male and female factors. Male infertility is any health issue in a man that lowers the chances of his female partner getting pregnant. Reasons for male infertility can generally be categorized into three groups: medical causes such as varicocele, infections, hormone imbalance, prior surgery, etc. Environmental causes like overexposure to toxic factors, environmental factors including chemicals, heat and toxins that can lead to a reduction in sperm function and production. Finally, overall health, lifestyle, or other causes can be the source of the problem, including alcohol intake, illicit drug use, tobacco smoking, stress, being overweight. The diagnostic process of male infertility includes a general physical examination and medical history, the examination of the patient's genitals and research on the patient's medical history, trying to identify those elements that might affect fertility, such as inherited conditions, chronic health problems, other illnesses, prior surgeries, etc. A patient is considered to have a low sperm count if he presents fewer than 15 million sperm per millimeter, while normal densities go from 15 million to greater than 200 million sperm per millimeter of semen. Some of these tests that are performed are scrotal ultrasound, hormone testing, testicular biopsy. When sperm concentration is extremely low, genetic causes could be involved, and here is where telomere testing plays a role, as you will see in the following slides. Telomeres seem to take up specific non-random positions inside the nucleus of sperm cells. Additionally, the idea that every chromosome has a particular position known as chromosome territory has been well accepted in the latest research. Dynamic length during spermatogenesis has been described by several researchers. In this process, telomeres shorten during the early stages of sperm differentiation, and by the end of the process, they elongate again, reaching even longer sizes than their initial length. Telomeres also play a role in chromosome recognition during cell division. They regulate recombination of homologous chromosomes and seem to be the first structure from the paternal nuclear genome to respond to signals after fertilization, leading this process to pronucleus formation. Different studies have shown that alterations in telomere length can lead to errors during programmed cell death, also having an impact on sperm maturation. Following this line, shorter telomeres in spermatozoids might be a leading cause of aneuploidy pathology related to reduction in fertility and recurrent pregnancy loss. Telomere length can be used as a biomarker for male infertility. Standard diagnostic procedures are often insufficient to explain the causes of infertility. New biomarkers with better clinical application are being investigated. Sperm telomere length testing is a fast and inexpensive form of sperm analysis, positioning this alternative as the next male infertility biomarker. If you want to study or want to know more about the correlation between male infertility and telomere length, here we provide you with a comprehensive list of publications. From here, we will describe how to implement our TAT test in your clinical practice. Our TAT should be considered a risk stratification biomarker. Even when a single measurement gives us valuable information about where our patient stands at the moment, the real value of the test comes from making several measurements that will produce longitudinal data. This allows us to know how a patient is evolving, how the personalized program that you have designed for him or her is impacting their telomere adhesion rate, or if you need to modify the program or add any different actions. What we recommend is to do the TAT prior to the beginning of the program to get a baseline, and then retest your patient after at least 1 year. Repeated annually, it is like a report card of whether lifestyle programs are working or not, indicating whether your patients are aging at a normal pace, at an accelerated pace, or ideally at a slower pace due to the efforts that they make to take care of themselves, like working out, eating right, stop smoking, managing stress, etc. If we see that this is not the case, we can adjust and take measures. This is a results report example. Here we provide you with the median telomere length, and this value is represented also in the graph that describes the different percentiles of the demographic distribution of telomere length. We also provide you with the 20 percentiles, which represents the percentage of short telomeres that your patient has. If your patient, our test, as I said before, is set up to be longitudinal. If your patient has done the test more than once, all the results are shown in the report. People have the opportunity to complete an anonymous health questionnaire online, and if the patient reports having any of the protective or risk factors that impact telomere length, these factors will appear highlighted in blue in the report. This slide shows the evolution over time of different technologies developed to measure telomere length. Our TAT is the most recent, technologically advanced, and clinically validated, which allow us to obtain not only the average telomere length, but also many other parameters, like the median telomere length and the percentage of short telomeres. These provide clinically relevant information about where your patients stand in their aging process and the likelihood of having age-related diseases. No other technique can provide all of this information with such accuracy as our TAT can. When compared to the other two most commonly used methods for telomere measurement, qPCR and flowfish, TAT is the only one that meets all three characteristics required for being a clinically useful test. Scalability, that allows us to process a great number of samples at the same time, which decreases the unit cost of the test, making it affordable for the patients. Total reliability, as our test is reproducible and accurate, granted by our strict quality program. And the clinical value we have talked about before, that only a clinically validated test can provide. This graphic summarizes all these attributes of our test. It allows multiple samples to be processed and to obtain median telomere length and percentage of short telomeres from single cells, not only from population of cells. TAT is a cutting-edge diagnostic test that looks at your aging process at the cellular level. It's possibly the best overall biomarker of health that exists. It is a simple test, as no special preparation or fasting for blood collection is required. Only one tube with 10 millimeters of blood is needed. We strongly recommend patients to complete the online questionnaire, as this will facilitate the interpretation of the results. The results report will be delivered to the physician in approximately 2 to 3 weeks, depending on the country of origin and shipping time for samples. We can provide this report in several different languages. Now we'll have a short break to allow a few minutes for people to ask any questions. I hope you found this presentation useful and helpful, and thank you very much for your attention.